Dear Chairman, ladies and gentlemen, it's my pleasure to take this opportunity to present our work in the Conference of International Research Awards on Oncology and Cancer Research. I'm Dr. Wang Meiyue from the Department of Physiology and a staff member of Professor Cao's Laboratory, Shanxi Medical University, in Shanxi Province, China. I will present for Professor Cao China. He is unavailable in recent couples of days. Our work is entitled High Expression of Interleukin-2 Receptor Subunit Gamma Reveals Poor Prognosis in Human Gastric Cancer. In the following presentation, I will name the keyword interleukin-2 receptor subunit gamma, abbreviated as IL2RG. First, I will introduce the incidence and mortality rate of gastric cancer. The incidence and mortality rate of gastric cancer is very different in the world. Geographical differentiation is one of the most distinctive characteristics of gastric cancer. Gastric cancer incidence is different concerning sex and geographical variability. Men are 2 to 3 times more susceptible than women. The incidence displays huge geographical diversity. Areas with the highest probability for gastric cancer development encompass regions like Central and South America, Western Europe, and East Asia, especially in China and Japan. This figure shows that the estimated age-standardized incidence in Asia-Pacific region for gastric cancer in 2018, both sexes or ages. From this picture, we can see that 5 countries in Asia-Pacific region with the highest age-standardized rates of gastric cancer incidence were South Korea, Mongolia, Japan, China, and Bhutan, respectively. Gastric cancer remains one of the most common and deadly cancers worldwide, with a mortality rate of the 3rd place and a 5-year survival rate of less than 30%. Countries with high gastric cancer mortality are mainly distributed in Asia, Latin America, and Europe. This figure shows the estimated age-standardized mortality rate in Asia-Pacific region for gastric cancer in 2018, both sexes or ages. 5 countries with the highest standard mortality rate from the gastric cancer in the Asia-Pacific region were Mongolia, Bhutan, China, Kyrgyzstan, and Tajikistan. Next, let's discuss what's the challenges in the treatment of gastric cancer. Although current clinical diagnosis and treatment have been developing, gastric cancer patients are often diagnosed in the late stage, while metastasis and recurrence may occur after radical excision in the earlier stage. The pathogenesis of gastric cancer is not fully clear. This situation leads to difficulties in earlier diagnosis, treatment, and prognosis. Precision medicine for gastric cancer is still an unsolved issue because most available target drugs are not specifically designed for gastric cancer. Exploring novel signaling molecules with target value for gastric cancer is in urgent need. These injured gastric cancer patients were from Shanxi Provincial People's Hospital. The gastric cancer tissues and paired adjacent to normal tissues were harvested during surgeries. These tissues were divided into 6 subgroups: male tumor, female tumor, mixed tumor, female normal, male normal, and mixed normal groups. All sequencing samples were compared on the expression level of transcripts per million. Figure 1 shows the distribution of expression level among all samples, was consistent, which met the requirement for differentially expressed genes analysis. Next, we identified the differentially expressed genes. Figures 2A through C are the volcano plots of the top 100 annotated differentially expressed genes, respectively in female, male, and mixed gastric cancer tissues. Blue dots indicate the downregulated genes, and red dots indicate the upregulated genes. From the figures, we can conclude that compared with respective normal gastric tissues, female, male, and mixed gastric cancer showed 1,057, 200, and 710 differentially expressed genes, respectively. Figures 2D through F are the height maps exhibiting the expression levels of differentially expressed genes, respectively for female, male, and mixed gastric cancer and non-gastric cancer gastric tissues. Line represents different genes and columns represent samples. Red means upregulated expression and blue means downregulated expression. Then we demonstrated the common and unique genes among these tissue subgroups. Cross-comparison of differentially expressed genes were performed for female, male, and mixed subgroups. Figure 3A shows that there were 49 overlapping differentially expressed genes among these three groups. The biological processes associated with differentially expressed genes were determined by KEGG pathway enrichment analysis. Figure 3B shows that cytokine receptor interaction was significantly enriched in both female tumor group and mixed tumor group. As a result, we focus on this pathway. In order to improve the accuracy of the comparison results of differentially expressed genes, the regulation information of transcription factor target and microRNA target, which had been verified by experiments, was collected. Results shows that based on the 8,427 pairs of transcription factor target regulation information in thrust database, searching differentially expressed gene regulating transcription factor, 44 pairs of transcription factor DEG regulation relationships were constructed, involving 14 DEGs and 38 transcription factors. Figure 4A shows these 44 pairs of regulation relationships. Based on the 8,157 pairs of microRNA regulation information in microRNA target interactions database, searching DEG regulating microRNA, 46 pairs of microRNA DEG regulation relationships were constructed, involving 14 differentially expressed genes and 40 microRNAs. Figure 4B shows these 46 pairs of regulation relationships. In addition, we analyzed the association of IL-2RG upregulation with gastric cancer progression and prognosis. Figure 5 shows that based on the differentially expressed genes identified in the gastric cancer and non-gastric cancer tissues, KEGG pathway enrichment analysis shows that IL-2RG was enriched in the cycle-time receptor interaction pathways and was significantly upregulated in gastric cancer. Figure 6a shows that IL-2RG gene expression level was significantly elevated in tumor tissues compared to the precancerous tissues. Figure 6b exhibits the correlation of the expression level of IL-2RG and the cancer pathological stage. The IL-2RG level was gradually higher with the prolongation of gastric cancer pathological stages. It can be seen in Figure 7c that IL-2RG was found highly expressed in gastric cancer samples compared with normal samples in GSE 29272 dataset. Figure 6d through F demonstrated that the overexpression of IL-2RG was associated with poor overall survival. Besides, to validate the IL-2RG abundance in gastric cancer tissue and gastric cancer cell line, we performed Western blotting and real-time qPCR. Figure 7a and 7b show that the mRNA levels of IL-2RG were much higher in both human gastric cancer cell lines and gastric cancer tissues, compared respectively with normal gastric epithelial cells and precancerous gastric tissues, as verified with real-time qPCR analysis. Consistent with the mRNA levels, Western blotting results in figures 7c through F show that IL-2RG expression levels were also significantly elevated both in human gastric cancer cell lines and gastric cancer tissues, compared respectively with normal gastric epithelial cells and precancerous gastric tissues. Finally, we found a small molecule named risotavir, or TAK242, might be an inhibitor of IL-2RG. It can be seen in Figure 7c that the asparagine 44 of IL-2RG is an important site to interact with risotavir. This molecular docking result suggests a way to find IL-2RG inhibitors, which might have therapeutic potential for gastric cancer. In addition, this work demonstrated that first, IL-2RG is highly expressed in both gastric cancer tissues and gastric cancer cell lines. Second, high expression of IL-2RG is associated with poor overall survival of gastric cancer. IL-2RG may be a proto-oncogene of gastric cancer and may serve as a biomarker or drug target for gastric cancer. Third, molecular docking showed that risotavir TAK242 might be an inhibitor of IL-2RG. Finally, pathway enrichment analysis allows a better understanding for gastric cancer. Gastric cancer pathology and may help to find rational target genes for gastric cancer therapies. We would like to acknowledge the National Natural Science Foundation of China Shanxi 1331 project key subjects construction and the Journal of Oncology for supporting this study. This is our article that can be found in this address. Thank you so much for your attention.